So welcome to the seminar of the SONY Astani Department of Civil and Environmental Engineering. Today our speaker is Rebecca Neumann. Let me tell you a bit about Rebecca. I believe she was raised in New Mexico and Colorado, and then found her way to Texas, where she actually got her Bachelor of Science at Rice University. And she started a streak of success by graduating magna cum laude. And that's a feature I've noticed in Rebecca. She was awarded so many fellowships and so on that she had to decline fellowships. She could not take all the awards she received. And it's a long list, but let me tell you a few of her achievements here. So after Rice, she went to MIT, and where she got, I believe, her PhD in October 2009. Now she's at Harvard, where she is a postdoc, a postdoctoral fellow. Actually, she's a NOAA, the National Oceanic and Atmospheric Administration, Climate and Global Change postdoctoral fellow. She's in the Department of Organismic and Evolutionary Biology at Harvard University. So she received so many awards that I cannot give them all of them, but let me point. I saw that she was a National Science Foundation Graduate Research Fellowship, a very, very prestigious fellowship from the National Science Foundations. She was also an MIT Presidential Fellow. She received, she was, I can't list all them all. Basically, she had so many awards that she could not accept them all. So we are very privileged today to have Rebecca to address us, and without saying further, for more, Rebecca, the floor is yours. So you want to click? You have a pocket, or you can click it?
Yeah.
You can hold it. So. Okay. Thanks, Rebecca.
Thank you. Thanks. Okay. All right. Thank you, Dr. Varday. So I'm going to speak today primarily about my PhD thesis work, which focused on the arsenic contamination problem over in Bangladesh. So the story of arsenic contamination in Bangladesh really began back in the 1970s when a number of aid organizations came into the country with the goal of reducing the high rate of waterborne diseases. And they actually successfully accomplished this by moving the country off of microbially contaminated surface water and onto groundwater. The effort was so successful that the aquifer is now the primary source of both drinking and irrigation water within the country. Unfortunately, after this transition occurred, it was discovered that the aquifer in Bangladesh is severely contaminated with geogenic or naturally occurring arsenic. This means millions of Bangladeshis are drinking water with unsafe levels of arsenic, and this water is used to irrigate a significant number of rice fields, which then does increase the arsenic content of the rice grains. So ingestion of arsenic results in arsenicosis, which manifests itself externally with these types of skin lesions. And internally, it causes a number of different cancers. So it's estimated that in Bangladesh, the arsenic exposure results in about 2 million cases of arsenic poisoning each year and about 3,000 fatalities each year. The general consensus is that the arsenic that we see within the groundwater, it was mobilized off the soil and sediments under reducing conditions. So microbes within the subsurface oxidize organic carbon, and then in the absence of oxygen, they use arsenic-bearing iron minerals, or perhaps even arsenic itself, as their electron acceptor. And if they use either of these as their electron acceptor, it ends up moving arsenic off of the soil and sediments and into the groundwater. So although there's agreement about this process in general, some key questions remain, including what I'm going to touch on today, which is the source of this organic carbon fueling microbial respiration and therefore responsible for arsenic mobilization. This is a brief outline of the talk, so I'm going to dive in and show you what arsenic concentration patterns look like in the aquifer in our study area. I'm going to present our hypotheses, explaining why we think arsenic has the patterns that it does, and then step through some data which support our hypotheses. And then what I'm actually going to do is spend a fair amount of time looking at rice fields, because it turns out that they play a pretty interesting role within the contamination problem. And then after that, I'm going to pull everything together, conclude, and step through some of the solutions for providing the country with clean drinking water that come out from the work that we did in Bangladesh. And then I just want to briefly switch directions a bit and kind of touch on where I see my research heading in the future. So arsenic in our study area has a distinct shape with depth. So concentrations are low in the shallow and deep part of the aquifer, and then reach a maximum at roughly 30 meters, which happens to be the same depth from which a majority of the drinking and irrigation wells are withdrawing water. And this distinct profile is not explained by solid phase arsenic concentration. So we don't see a real enrichment of arsenic in the same area where we see high levels of arsenic. So we hypothesized that this pattern was instead due to the origin of the groundwater. So we suspected that water which was low in arsenic evolved from a different recharge source than the water which is high in arsenic. So,
I'm sorry.
Yeah.
A couple slides ago, there, you do have that one outlying data point right at what is the deal?
Yeah, so there's just, I'd say it's kind of an anomalous one outlying data point. So maybe we hit some nodule which had, was a mineral which had a lot of arsenic in it. But, if you look kind of throughout this whole depth, concentrations are low. And actually kind of the crustal average for arsenic is up around 8 milligrams per kilogram. So in general, arsenic within the sediment in Bangladesh isn't anonymously high compared to what you would see probably if we even measured arsenic out here in the soil on campus. And so I guess, which is typical in most research endeavors, you get this kind of one thing that maybe complicates the story a little bit. But I think even if we look at other researchers' field sites, it's a similar thing, that there's no real clear increase of arsenic at this peak found throughout the country. So this is the water budget for our study area, and it shows that irrigation pumping is the largest discharge source from the aquifer, while rice fields and ponds are the primary recharge sources, each contributing about roughly equal amounts of water. And then we have rivers, which act as both a discharge and recharge source depending on the time of the year. What I want to point out is that these primary recharge sources, so the rice fields and the ponds, these are actually man-made. So back again in the 1970s, when all these wells were installed around the country, it suddenly gave farmers access to groundwater as a source of irrigation water. This then allowed them to cultivate a dry season rice crop, which we call boro rice. And you'll see that the area under groundwater irrigation and experiencing boro rice cultivation have both continually increased since their introduction into the country. This has had a profound impact on the landscape. So in our study area, groundwater irrigated rice fields now cover up to 40% of the land area. This agricultural boom was accompanied by a growth in population. And this growth in population in turn caused an increase in the number of these man-made or dug ponds. So let me explain the link between those two. This is a picture of one of these man-made ponds, and you'll notice that it surrounds a village which is raised up above the rest of the landscape. And this is done because each year in Bangladesh the landscape is flooded with water, and so they need to build villages and roads up above the level of these floods. And they do this by digging material up from one location and then transferring it to another. And the hole from which they obtained the material fills with water during the monsoon season and then sits on the landscape as a pond during the dry season, recharging the aquifer. So as the population increased, the number of these man-made villages had to increase, which caused an increase in the number of these man-made ponds. So much of the water budget for the aquifer in Bangladesh is largely controlled by the changes that humans have made to the landscape over the past roughly 50 years. So returning to the water origin hypothesis, it's really supported by a few lines of evidence. So the first is when we looked at the radiocarbon age of inorganic carbon pumped up from this 30-meter aquifer depth, we found that the carbon was much younger than the age of the aquifer itself. So this meant that the organic carbon fueling microbial respiration could not have been co-deposited back when the aquifer was formed. Instead, this carbon had to be transported into the subsurface, so transported from the surface with the recharge water into the subsurface. So highlighting the importance of recharge, bringing organic carbon into the system. In addition, when we looked at conservative tracers, we found that the water at the arsenic peak was distinctly different than the water found within the rest of the aquifer. So what I'm showing here are data for deuterium and chloride. These are considered conservative tracers. So they don't interact with the solid phase and they don't undergo any type of chemical reaction. So they really act as a type of chemical fingerprint for different water types within the aquifer. And when we compared this measured profile in the aquifer to that which we found in water coming out from the bottom of a pond and then water within the subsoil of a rice field, we immediately see that the pond water has the same isotopically light and low chloride concentrations that we see at the arsenic peak. And in fact, we can get a good match to the entire measured profile in the aquifer if we assume the aquifer is dominated by 3 water types. So water evolving from ponds, water evolving from rice fields, and then old resident groundwater which existed in the aquifer prior to the development of rice fields and ponds. And we decided that our deepest aquifer wells were the best representation for this pre-development water. So if we treat these 3 representative water types as end members, mix them together using the proportions shown here, we predict a concentration profile for the aquifer, which is this black line, which nicely matches that which we measured in the aquifer. So again, this black line shows the concentration of deuterium and chloride that we would expect to see in the aquifer if the shallow depths are dominated by water evolving out of rice fields, if the middle depth of the aquifer is dominated by water evolving from ponds, and then if the deepest depths of the aquifer is composed of this old resident groundwater. So this nice match between the measured and the modeled concentrations demonstrate that recharge is distinctly layered in the aquifer, and that it's this pond recharge which is associated with the elevated levels of arsenic that we see in the aquifer. And this layering of recharge in the aquifer is physically explained by the constraints of our water budget. So again, irrigation pumping is the largest discharge source. So we expect most of the water in the aquifer is going to be converging on the depth of the irrigation wells. We then have rice fields and ponds contributing roughly equal amounts of water, but rice fields cover close to 40% of the land area and ponds cover only 10%. This means that when the pond water enters the subsurface, it has to spread out laterally, and it spreads out laterally beneath that of the rice field recharge. So if we were at a location relatively far from the influence of a river, we would expect to see this type of flow system being established within the aquifer. And if we were to look at a profile from beneath or adjacent to a rice field, we would see rice recharge dominating the shallow depths, pond recharge dominating the middle depths, and then old resident groundwater within the deepest depths. And we're particularly interested in locations beneath and adjacent to rice fields because this is where we've installed our research observation wells. So this is the location from which we've gotten the arsenic data, deuterium data, and chloride data shown in the presentation. So the constraints of our water budget and our isotopic and other conservative tracers suggest that the pond recharge is evolving into the high arsenic groundwater and the rice field recharge is evolving into the low arsenic groundwater. So the question is why? Why are the different recharge sources behaving differently? To answer that question, we moved to the recharge sources themselves. So now we're going to be focusing just on the top 3 meters of the subsurface. So our hypothesis was that the different behaviors was due to the biological availability of the organic carbon brought into the subsurface with the recharging water. So again, remember, we need to have organic carbon to stimulate microbial respiration and get the arsenic mobilization. So to test this hypothesis, we collected pore water from the sediments of 2 different ponds and from a rice field subsoil. We filtered this water, we sealed it in the field, we brought it back to the lab, inoculated the samples with unfiltered water, and then exposed the samples to the atmosphere and monitored the organic carbon concentrations with time. We basically found no loss of organic carbon within the rice field subsample, whereas in the 2 pond samples we saw a loss of about 0.5 millimole of organic carbon. So the experiment shows us that the microbes within the inoculum, which were applied to the rice field sample, they were not able to oxidize the organic carbon in that water sample, where the microbes applied to the pond sample were able to oxidize a portion of that organic carbon. So the experiment showed us that not only does the rice field recharge have less organic carbon in it than the recharge coming from ponds, but essentially none of this carbon is biologically available. In contrast, the carbon coming from the pond has a portion which is biologically available, which means that the pond recharge can stimulate microbial respiration and mobilize arsenic off the soil and sediments. And in agreement with these results, when we look at arsenic concentrations within the pore water of a pond sediment, we find a continual increase with depth. And this is a trend that we believe continues as this pond water is pulled further into the subsurface down towards the depth of the irrigation wells. In contrast, when we look at pore water concentrations of arsenic within a rice field subsoil, we see no clear trend with depth. And when we consider that the rice fields are actually irrigated with arsenic contaminated water, we immediately see that the arsenic concentrations within the subsoil are more than an order of magnitude less than that in the irrigation water applied to the field. So the question is now, I think, why are the rice fields producing low arsenic groundwater? So it's counterintuitive and truthfully exactly opposite to what we expected when we began this work, because we know rice fields are organic rich. For example, they're a huge methane source to the environment. So there is biologically available organic carbon within the rice fields. In addition, in Bangladesh, these fields are being irrigated with arsenic contaminated water. So why is the recharge coming from the rice fields low in arsenic? To answer that question, I'm going to step through a pretty detailed hydrologic and chemical investigation that I conducted on the rice fields. So I ended up monitoring water levels in 4 fields of various sizes, and then I intensively studied one of these fields using a number of different methods. And I'm going to step through in this presentation results from our 3 season-long tracer tests, from a sensor transect, a hydrologic sensor transect, that we installed from the middle of the rice field into the bund. And the bund is this raised boundary around the edge of the field. And from soil cores, which we collected both within the bund and within the planted portion of the rice field.
Is this a plan view? What am I looking at?
Okay, so this is a map of Bangladesh.
Yeah.
This is our field site. So then this is a zoomed in view of the field site. So these are the 4 fields I studied. And then this red line here is supposed to show then this zoomed in view here. And so this is field A, and then this is field A here. And I'm showing where within the field I conducted 3 tracer tests, and spatially where my sensors were and the soil cores. So it is just looking a top-down view onto the rice field. So all of the data that we collected in the rice field were consistent with the fact that the flow patterns in the field are controlled by cracks, and then by the field preparation practices of the farmers. So cracks form in the rice field each year at the end of the irrigation season when the soil dries out. And these cracks will remain open and active even after the field is resubmerged with water. So in order to seal up these cracks, the farmers plow their fields in water-saturated conditions. And this breaks soil aggregates up into finer soil particles that can then fill in the cracks and void spaces when they settle out of the water column. Notably, the farmers do not plow through the bunds, which again are these raised boundaries around the edge of the field. And the bunds aren't plowed primarily because they follow property boundaries. This is an X-ray of the soil cores that we took, and it shows that because the bund is not plowed, it's full of cracks and void spaces, while the soil from the planted portion of the field, which is plowed, lacks cracks. So ultimately, plowing creates a surface layer of soil, which we call as a plow pan. And because this plow pan lacks cracks, it has a lower effective hydraulic conductivity than the soil found within the bund or the subsoil found beneath the plow pan. And some researchers in the 1980s, named Walker and Rushton, they developed a steady-state numerical model for just a generic rice field. And this model showed that the conductivity difference between the plow pan and the bund allows surface water in the field to flow into the bund, down the bund, into the subsurface of the field. And then the conductivity difference between the plow pan and the subsoil allows for the development of an unsaturated zone, even when the surface of the field is flooded with water. So all of our data are consistent with this general flow pattern, but also highlight that the field hydrology is more dynamic and complex than these generic steady-state models had predicted. So the 3 season-long tracer tests that we conducted allowed us to visualize flow paths of water which managed to infiltrate vertically through the plow pan into the subsurface of the field. So in each of these 3 locations, we released a solution of bromide into the rice field using one of these types of concrete rings. When the solution had fully infiltrated, we removed the ring and then left the field to the management of the rice farmer. We returned after the rice had been harvested, dug a trench at those 3 locations, and then sprayed the face of a trench with a chemical that reacted with bromide to form a dark Prussian blue color. So the experiment showed us that water in the surface of the rice field was able to reach depths of at least 150 centimeters by flowing through cracks and void spaces within the subsoil of the field. In contrast, water which remained within the soil matrix only managed to reach a depth of about 40 centimeters during the irrigation season. And since we released the tracer at the start of the season, visualized it at the end of the season, the experiment shows us that the only water which is able to reach and recharge the arsenic-contaminated aquifer is water which enters one of these preferential flow channels. So any water which remains in the soil matrix never reaches the aquifer during that irrigation season. Now the data from our 3 season-long, I already talked about the tracer test. So the data from our sensor transect, it allowed us to visualize flow paths across the entire transect of the rice field. And our transect was composed of 9 tensiometers, which measured both water pressure and water tension, and we collected data each hour from this transect during the irrigation season. So, due to the dynamic nature of the transect data, it's best viewed as a movie. So what I'm going to do is have the movie fade in here behind the cartoon, so you have an idea of what you're looking at. Maybe I'll do it one more time. So these black dots mark the location of the tensiometer measurements, and the y-axis here marks the location of the bund. So what I'm showing are linearly interpolated hydraulic head contours. So hydraulic head is energy per unit weight of water, and it's a function of both pressure and elevation. So we have pressure measurements from each of these 9 tensiometers. We combine that with the elevation from which the measurement was made to calculate a head value. And these contour lines are then the linear interpolation of those head values. So water flow is perpendicular to the contours in the direction of decreasing head. And the value of the head contour line is marked by its color, according to that laid out here in the color bar. This blue bar marks the height of ponded water sitting in the rice field, and you'll see this bar move up and down as irrigation water is added and lost from the field. And then finally, you'll see one of these black dots turn white when that tensiometer measures negative pressures, which signifies unsaturated conditions. And a black line will show up which marks the interface between the saturated and the unsaturated zone. And the location of that black line is simply calculated from the contours. So the unsaturated zone is the region where the head contour value is less than the corresponding elevation, which signifies negative pressures and therefore unsaturated conditions. So I'm going to start the movie. And you'll notice that during the entire irrigation season, water flow in the middle part of the rice field is vertically downwards through the plow pan into the subsurface of the field. And the tightness of these horizontal contours is due to the low effective hydraulic conductivity of the plow pan. When water is added to the field, it flows from the surface of the field through the bund into the subsurface of the field. And we'll see this occur again. Irrigation water is added to the field and we get active bund flow. This compresses the unsaturated zone into the middle portion of the field. As the water level in the rice field decreases, bund flow also decreases because there's less of the bund surface area covered by water. And when this happens, the unsaturated zone grows such that it eventually occupies the entire area beneath the rice field. As the farmer stops irrigating the field, the entire soil profile begins to desaturate. And this downward head gradient suggests that desaturation is due to drainage rather than due to evaporation. However, as we continue into the dry season, evaporation eventually causes the head gradient to switch directions, which is exactly what we're seeing occur right now. Evaporation then continues to dry out the surface of the field, causing the cracks, which I showed in the photograph earlier in the presentation. These cracks break open the plow pan so that when we get these early monsoon rainwaters, the water quickly bypasses the soil matrix and recharges the deeper soil layers. When we have no longer an active plow pan, we do not get bund flow even when there's ponded water sitting on the field. So in case you didn't follow every part of the movie, I'm going to summarize the main points with contours from 3 time periods. So immediately after and during an irrigation event, water flows from the surface of the field into the bund, down the bund, into the subsurface of the field. And this active bund flow compresses the unsaturated zone into the middle portion of the field. As the water level in the field decreases and we have less area of the bund covered by water, bund flow also decreases, and then the unsaturated zone grows such that it eventually occupies the entire area underneath the field. We then move into the dry season, and soil drying causes the cracks, which break open the plow pan. And with a broken plow pan, we no longer get bund flow even when there's ponded water sitting on the field. In addition, these cracks allow this early monsoon rainwater to bypass the soil matrix and quickly recharge the deeper soil layers. So our data show us that the rice field is gaining water from irrigation and rain and losing water to evaporation, preferential flow through cracks and void spaces within the subsoil, infiltration into the soil matrix, and then loss out the bund. Where B here is the bund flux, which is bund flow normalized by A, which is the area of the rice field. And bund flow is a function of the area of the bund covered by water. So H sub P, the height of ponded water, multiplied by P, which is the perimeter of the rice field. So we quantified all of these fluxes for 2 different irrigation seasons and determined that of all the irrigation water applied to the field, about half of it flows out the bund. But when we consider just the water, which has the potential to recharge the arsenic-contaminated aquifer, we find that about 80% of this water is flowing through the bund. So we confirmed the importance of bund loss by looking at the water level data from the other fields in our study area. So we found that when the bund was covered by water, the rate of water loss was linearly related to the perimeter to area ratio of the field. So smaller fields have more bund exposure per unit area field and therefore lose proportionately more water out their bunds. So this difference in bund exposure resulted in a relatively strong relationship between the total amount of irrigation water applied to the field and the size of the field. So what I ended up doing was using this relationship to predict how much a field of a given size will likely receive in irrigation water, and I subtracted from that total amount all of the losses other than that due to bund flow. That gave me a function of the fraction of water lost down the bund as a function of the perimeter to area ratio. So this shows us that small fields are losing almost all their water out the bund, and large fields lose almost no water out their bund. When I compare this to data from different field sites, we get a pretty nice match. So this is for our intensively studied field site that I've showed data for, and these two other field sites are the only two sites published in the literature for which I could calculate a perimeter to area ratio. Both of these rice fields are located in the Philippines. So this suggests that this is a somewhat universal relationship, and it shows us that farmers could actually save a fair amount of water, especially farmers that own small fields, if they could reduce bund loss. And they could do this by extending the plow pan beneath the bund, so by plowing through the bunds and rebuilding them each year. A pretty easy and simple way to save on water. But if we return to the arsenic contamination problem in Bangladesh, we're interested in the water which is reaching and recharging the aquifer. So this relationship shows us that for small fields, this water is going to be flowing through the bund, and for large fields, this water is going to be flowing through the cracks and void spaces within the subsoil of the rice field. In our study area, the average perimeter to area ratio is at 0.12, which suggests that most of the irrigation return flow is flowing through the bund. Recall that for our intensively studied field site, when we consider just the water recharging into the aquifer, so not the total amount of water applied to the field, just the water reaching the aquifer, more than 80% of it had flowed through the bund. So this hydrologic investigation shows us that most of the rice field recharge in our study area is flowing through the bunds, and that this flow is not constant in time. So it's occurring immediately after an irrigation event when a larger surface area of the bund is covered by water. So this understanding of flow then helps us interpret the behavior of organic carbon and arsenic within the rice field. So when we looked at dissolved organic carbon concentrations in the surface water of the field, this is immediately after an irrigation event, we found that the concentration matched that measured in the irrigation water itself. We then found that the organic carbon concentration over the next couple of days essentially doubled. So as the water sat and interacted with the organic rich surface of the rice field, we get a doubling of organic carbon concentrations. When we look at dissolved organic carbon concentrations within the pore water of the bund, we see the concentrations match that which we measured immediately after irrigation, right? Which makes sense because this is the time period during which bund flow is occurring, during and immediately after an irrigation event. So these data show us that actually much of the organic carbon in the rice field, so this carbon which is responsible for all the methane generation that the rice fields are doing, it gets retained within the surface of the field. So this carbon is not getting transported deeper into the subsurface where it can mobilize arsenic. In addition, the carbon which does recharge into the aquifer is likely the same carbon which we pumped up out of the aquifer in the irrigation water. So this carbon is likely old and highly recalcitrant, explaining why when we looked for biologically available organic carbon within the pore water of the bund, we basically found none. We basically found all the carbon was highly recalcitrant. So this flow pattern helps explain why we aren't getting fresh arsenic mobilization off the soil and sediments as the rice field recharge moves through the subsurface into the aquifer. The question, though, is what's happening now to all of this arsenic that we're applying to the field in the irrigation water? So we tracked the fate of this irrigation arsenic as it was pumped out of the aquifer and deposited onto the rice fields. So when irrigation water is applied to the field, it usually enters the field in one location, which we call the irrigation inlet, and it slowly moves and spreads across the field over a course of a couple of hours. And during this time, the water is getting exposed to oxygen, which causes the formation of arsenic solids, which then settle out of the water column into the surface soils. In addition, this water is flowing through and interacting with these oxidized surface soils, which can act to sorb arsenic out of the water and onto the soil. So this is data collected by our colleagues at AVOG over in Switzerland. They showed that just as irrigation water moved from the inlet to the far corner of the field, we had about a factor of 4 decrease in the arsenic concentration in the surface water. And then we find that about 48 hours after an irrigation event, arsenic in the surface water is essentially below detection limits. Yes?
How much time did it take the water to flow that distance?
It takes like 2 hours or something on that order. And I think this sampling that she did was within a couple hours after the irrigation event. So we expect that much of this irrigation arsenic is going to settle into the surface soils of the field. But we also know that bund flow is actively occurring immediately after the irrigation event. So we expect some of this arsenic is also going to enter the bund. And we care about the partitioning of these two because arsenic, which enters into the surface soil, can then enter the rice crop and impact the arsenic content of the rice grains. Whereas arsenic, which enters into the bund, is not going to impact the crop, but then has the opportunity to recycle back and recharge into the aquifer. So we determined the flux of arsenic moving into the bund by combining data that we had on the water flow into the bund with the measured concentrations of arsenic within the surface water of the field. And by combining these two together, we determined the flux of arsenic entering the bund. And again, this flux is spatially variable because of that decreasing trend in concentrations of arsenic that we see immediately after the irrigation event. So we had flux estimates for 2 different irrigation events on our studied field, and we scaled that up to the entire irrigation season using water level data that we collected for the entire season. And with that, we determined that about 1/3 of the arsenic which is applied to the field enters the bund, and the remainder settles into the surface soils of the field. The question is how much of this arsenic that enters the bund then reaches and recharges the aquifer. To look at that, we focused on arsenic concentrations in the water flowing from the bund into the aquifer. And we really focused on arsenic measured in a relatively large, so 12 shallow aquifer wells installed directly beneath and adjacent to the rice fields. So we combined this measured concentration then with the volume of water moving through the bund to get the flux of arsenic moving from the bund into the aquifer. And we just made sure that this estimated flux made sense with respect to the amount of arsenic that we estimated was entering the bund, and then measured concentrations of arsenic in the soils of the bund. And with that, we determined that very little of the arsenic is actually making its way back into the aquifer. So most of this irrigation arsenic is being retained within the soils of the bund and the surface soils of the rice field. This means that irrigation pumping is removing arsenic from the aquifer each irrigation season. Fortunately, some of this arsenic which is retained within the soils is lost from the system during the monsoon season. So we have floodwaters sitting on top of the rice fields, and we get reducing conditions at the floodwater rice field interface. This causes the arsenic solids which had formed during the irrigation season to dissolve. The dissolved arsenic then diffuses up into the floodwaters and is flushed from the system when the floodwaters move out to the Bay of Bengal. So this is data again collected by our colleagues at AVOG, and it shows measured concentrations of arsenic in a floodwater column on top of a rice field. We also lose arsenic from the system as the floodwaters move across the landscape and erode the soils within the bund. So using data that we collected on bund erosion and then data our colleagues collected on loss of arsenic into the floodwaters, we calculate that about half of the irrigation arsenic applied to the field is then lost from the system during the monsoon season. So this results in a steady accumulation of arsenic within the soils of the bund and the soils of the rice field. So in some ways, irrigation of arsenic is actually acting like an unintentional pump and treat system for the aquifer. So arsenic is pumped up out of the aquifer in the irrigation of water and applied to the rice fields. Much of this arsenic is retained within the soils of the field, and then about half of it is then flushed from the system during the monsoon season. So what I'd like to do now is just pull everything together and kind of step through what we see as the story of arsenic contamination in Bangladesh. So we have ponds, which are pulling biologically available organic carbon into the subsurface. This carbon stimulates microbial respiration, which then acts to mobilize arsenic off the soil and sediments as this water is pulled down towards the depth of the irrigation wells. This arsenic is then pumped up out of the irrigation in the irrigation wells and applied to the rice fields. Much of this arsenic is then retained within the surface soils of the field, and half of it is lost then into the monsoon floodwaters. And because of the flow patterns in the rice fields, the recharge coming in from the rice fields is not bringing with it biologically available organic carbon. So we are not stimulating microbial respiration, and arsenic concentrations within the recharge of the rice fields remains low in arsenic. So these results suggest that there are two different possibilities for providing the country with access to arsenic-free groundwater. The first is to install deep drinking water wells, and this is actually a solution that's been put forward by a number of different researchers. Our results demonstrate that this solution is a good and sustainable solution as long as we don't also put irrigation wells deeper into the subsurface. So if we also drop irrigation wells, we're inevitably going to pull pond recharge down towards this depth, and we would contaminate these drinking water wells. Another possible solution is to put in shallow drinking water wells that actually capture this irrigation return flow from the rice fields. So this solution is cheaper. It's maybe $100 to put in one of these wells versus over $1,000 to put in one of these deep wells. But these shallow wells would be more susceptible to perhaps microbial contamination or any kind of management decisions the farmers make. So it's not a slam dunk solution, but perhaps one that's at least worth considering. Okay, so that summarizes the work that I did in Bangladesh, and what I'd like to do now is just shift directions a bit and talk about where I see research heading in the future, if you feel like we have enough time. Okay. So I'm really interested in environmental fluxes, so fluxes of water, nutrients, carbon, and contaminants through the interconnected environmental system. And I'm particularly interested in how agriculture can shape these fluxes and how these fluxes can influence agriculture. And I think that the interaction and feedback between these two are particularly important because of the implications that they have for human health. Specifically, as we move into the future, we have increased population density, altered climate conditions. There's going to be significant strains on our food and our water systems, and the two are really intimately intertwined. And I would like to know how the decisions that we make, be it about land use, resource management, or even emission of CO2 in the atmosphere. So how are these decisions going to impact the quality of our food and the quality of our water? And I think that the only way we're going to know that is by tracking these fluxes, because the fluxes are really the mechanism by which our decisions and the impacts of our decisions get propagated through this interconnected environmental system. And I think I'm really well poised to work at this intersection because of the training that I presented in this talk during my PhD in hydrology and biogeochemistry. And then the training that I'm currently receiving in my postdoc in plant physiological ecology. So I'm currently working with 2 plant biologists looking at how plant roots can modulate their hydraulic conductivity in response to environmental conditions. And the idea is to understand how these modulations in root conductivity influence the amount of water that plants are moving through the landscape. And my goal with this postdoc is really just to gain a better understanding of plants and how they work, and in particular how plant roots work. And also to gain access to the methods and techniques that are available for working with and studying with plants. And then I plan to take these knowledge and this skill with me into my future career, so I can really address what I think are the important questions that fall at this interdisciplinary interface. So I'm particularly interested in soils because they're the critical interface between the atmosphere and the surface and the land surface. And the minute that you start looking and thinking about soils, you can't ignore the fact that there are plants in the soils which are having a big impact on our water cycle, our nitrogen cycle, and just soil biogeochemistry. So if there's time, I thought I would just briefly step through 2 project ideas that I have. And these are just 2 of many ideas. The first begins to build upon my PhD thesis work, and it looks at how different rice field water management schemes may impact arsenic concentrations in rice, drinking water, and irrigation wells in Bangladesh. And this is of particular importance because the International Rice Research Institute is currently teaming up with local groups in Bangladesh to promote a new type of rice field management called alternate wetting and drying. And the purpose of this management scheme is to reduce irrigation use. So it's the same motivation that I had in my thesis, where I suggested that farmers could plow through and rebuild their bunds each year. However, I think that before any type of management scheme is promoted on a national level within the country of Bangladesh, we just need to recognize that the fields are part of a larger hydrologic and biogeochemical system. And so if we perturb one part of that system, we're going to have impacts elsewhere. In particular, if we reduce irrigation pumping and reduce the amount of recharge coming out from rice fields, we're going to increase the volume of the aquifer receiving recharge from ponds. And we know that it's pond recharge which is associated with the elevated levels of arsenic. So we're potentially going to increase the area of the aquifer experiencing arsenic contamination. This could contaminate currently clean drinking water wells, and it likely would also increase arsenic concentrations in irrigation wells, which then means that we are going to have impacts on the rice crops grown with this irrigation water. In addition, I think it's also important just to think about how these rice field management schemes may impact the solutions that people are putting forward for providing the country clean drinking water. So we want to make sure that any policy that we pursue or promote within the country ends up having a net positive impact on human health. So my plan would be to use field studies, laboratory experiments, and reactive transport modeling to evaluate the different management schemes so I can then make a recommendation about their use within the country. And then a second project begins to build upon my postdoctoral work, and that's looking at zinc availability and uptake by wheat when it's grown under elevated CO2. And so this is a project that I've developed after meeting and establishing a collaboration with a physician at the Harvard Medical School. And he's really spearheading a larger effort to look at the human health implications of altered nutrient quality in crops grown under elevated CO2. So here I'm showing data for wheat grain grown under about 2 times ambient CO2. And most of the macronutrients and all of the micronutrients decrease. And currently, I kind of picked up on this decrease in zinc because zinc deficiency is already a significant human health problem, so it weakens your immune system and makes you more susceptible to other diseases. And a 20% decrease in the content of wheat, which is a globally important crop, could potentially exacerbate an already significant human health problem. But to really get at the human health implications, we need to understand what we think zinc concentrations are going to look like in plants grown within different regions of the world, particularly within regions where zinc deficiency is already an issue. And so I think that the first step in getting at this is to look at the supply side of the equation. So how is elevated CO2 going to alter zinc solubility? And then how is it going to alter the transport of zinc to the root? And both of these factors are important because uptake of zinc by the root is a function of the dissolved concentration of zinc at the root surface. So what I'm showing here is a picture of the rhizosphere. It's the soil zone around roots. And it's supposed to represent something on the order of a couple millimeters. But what I want to highlight is that the system is not all that different from the one in Bangladesh. So in Bangladesh, we have irrigation pumping, pulling large amounts of water out of the subsurface. In the rhizosphere, we also have a well, it's the root itself, pumping water out of a very spatially distinct region within the soil. In Bangladesh, we had an input of organic carbon coming in from the ponds. In the rhizosphere, we also have an input of carbon coming from the root itself. And just like in Bangladesh, this carbon can stimulate microbial respiration, which is going to alter soil biogeochemistry. So under elevated CO2, we expect that the amount of carbon being put out by the root is going to increase, which is going to alter microbial activity and likely change zinc solubility. In addition, under elevated CO2, we expect transpiration rates are going to decrease, which means that the pumping rate of the root is going to decrease, which is going to alter flow patterns and water contents within the rhizosphere, which means that we're going to alter transport of zinc to the root surface. So my plan is to use laboratory experiments and numerical modeling to really study the processes and interactions within the rhizosphere that control zinc, and to think about and study how sensitive these are to elevated CO2. Ultimately, my goal is to create a mechanistic model of the rhizosphere that we can use to not only look at zinc, but look at other key nutrients such as nitrogen. And the idea is that with this model, we'll be able to test and develop ideas for properly managing the rhizosphere so we can end up increasing nutrient uptake and decreasing such things as like runoff of fertilizers into the greater rivers and groundwater, for example. So, okay, so that summarizes my work, and I just want to move back to Bangladesh and highlight all the people that made the work possible. We had a number of funding agencies, my thesis committee in particular, my advisor, Charles Harvey. And then I just want to pull out our collaborators over there at the Bangladesh University of Engineering and Technology, Borhan Badruzman and Ashraf Ali. So their work and support was really instrumental in the success of our program. I have a lot of people over in Bangladesh that lent me a hand, a number of researchers who came and helped me in the field, some helped me in the lab, and then just general intellectual support. Thank you. Happy to take questions. Thanks. Yeah.
You suggested sealing the bunds to prevent loss of water and influence this other stuff. Would that carry a risk of field salinization because it reduces the flow through?
So I guess I don't think so, because the water that is flowing through the bunds isn't actually moving through the rice field part itself. So it really is literally just kind of in terms of agriculture wasted water. So we still get this infiltration of water in the planted portion of the rice field, that slow infiltration through the plow pan into the soil matrix. But this bund flow, it really just kind of gets shunted out of the system immediately after irrigation. And I think issues of salinization and stuff, my understanding is they're not as big of an issue in rice agriculture, because I think you have kind of the field is continually flooded with water, so the evaporation isn't, my sense is that it's not kind of concentrating salts and solutions in the soil.
What kind of arsenic are we talking about? Arsenite or arsenic?
Okay, so in the aquifer itself, it's arsenite, so it's highly reduced arsenic. Well, it's not arsine, but it's highly reducing conditions within the aquifer, so all arsenic 3. And then what happens is when it gets pumped up out of the aquifer and then put onto the rice fields, it turns into arsenic 5. And in the kind of the shallow layers of the rice field, it, I think, remains arsenic 5 because the water stays pretty oxygenated. You have algae floating in there that are, so the water actually gets super saturated during the day. And then you have this kind of mucky soil layer beneath that, which is highly reducing. So that's where all the methane generation is happening. And I didn't specifically measure it, but I would guarantee that that's arsenic 3 down where we're getting methane generation. And then I guess as arsenic is then moving through the bunds, I don't think we're getting, I think it would probably stay that arsenic 5, but then any arsenic then which enters into the aquifer is going to get converted back to arsenic 3.
But you were talking about 1,000 micrograms per liter, so did you use total arsenic?
Yeah, so in the wells we're getting up to 1,000 micro. Yeah, so
Total.
yeah, so I tended to use unfiltered kind of total arsenic in my studies of the ponds and then in the rice fields because I really cared about kind of bulk movement of arsenic through the system. And then, but the data from the aquifer wells, so the deeper part, so including that 30-meter arsenic peak, we did speciate that. So we looked at arsenic 5, arsenic 3, and none of it was arsenic 5.
Okay, well, in the interest of time, I think we're going to have to conclude. I know you have more questions, but I'm looking at the clock, and I believe you have a very busy schedule today. But I think we should thank Rebecca for a very interesting seminar.
Okay.
Thank you.
Thank you. Thank you. Do you want to just ask your question offline?
That's okay.
Great job.
Okay.
You did a
Okay.
great job.
Thank you.